Hi, I am Peter Andrew, a PhD candidate at UC Davis working in the lab of Pam Line. Our lab studies the consequences of intoxication with a class of compounds known as organophosphates, a class which includes both pesticides and nerve agents. These compounds inhibit the enzyme acetylcholinesterase, resulting in excessive cholinergic signaling and ultimately seizures. So our lab is interested in studying the neuropathologies that are caused by such intoxications and ultimately developing therapeutic strategies to mitigate long-term consequences of organophosphate intoxication. My projects focus on a couple of these long-term consequences, notably neuroinflammation and the development of spontaneous recurrent seizures, and the relationship between these two consequences. So there is evidence from the broader seizure field that neuroinflammatory processes can exert epileptogenic effects. In other words, they can predispose an individual to the development of spontaneous recurrent seizures or acquired epilepsy. In this particular project, I aim to investigate the involvement of microglia, which are the brain's innate immune cells, and their activation following organophosphate intoxication ultimately to the development of spontaneous recurrent seizures. To do this, I provided an inhibitor of microglial activation, minocycline, to rats acutely intoxicated with the prototypical organophosphate diisopropyl fluorophosphate, or DFP. You can see here the experimental design and timeline for these projects. Animals were acutely intoxicated with DFP and then provided standard of care therapeutic countermeasures or standard of care plus daily minocycline injections. Minocycline treatment attenuated metrics of microglial activation following DFP intoxication, reducing the total density of microglial cells in the piriform cortex and amygdala at 7 days post-exposure. Similarly, at 28 days post-exposure, morphometric analysis of microglial populations demonstrates that minocycline treatment shifts populations towards a more ramified homeostatic morphology compared to those animals not treated with minocycline. Moreover, animals administered minocycline had a reduced incidence of spontaneous recurrent seizure development and overall had a reduced seizure burden compared to those animals not administered minocycline. In all, these data present pharmacologically attenuated microglial activation coincident with reduced incidence of spontaneous recurrent seizure development following acute DFP intoxication. Such findings suggest the epileptogenic effects of microglial activation following OP intoxication and advocate for further evaluation of the relationship between neuroinflammatory and epileptogenic processes across chemoconvulsant and neuroinflammation models. I wanted to thank ARCS for the continued support, and I look forward to seeing you all at the symposium in April.